Hello. So, we were talking about those enzymes on nickel, and these were basically some ancient enzymes. We call them also because in some cases these are very much useful for the primitive organisms. So, those primitive organisms, they basically rely on or live on some anaerobic atmosphere. That means, it is basically a CO2 rich environment. So, for that what we see here, that both carbon monoxide dehydrogenase and acetyl coenzyme synthase both are responsible for reduction of CO2 to CO, and that CO is subsequently transformed into acetyl coenzyme A. So, these basically are very important thing, particularly when our environment is anaerobic. So, we are not talking anything related to dioxygen environment. So, for that reason we can also consider all these reactions as anaerobic metabolism. And sometime, since we are talking or we are considering one carbon center, that means whether it is from the carbon monoxide or carbon dioxide or the acetyl function, because this acetyl function can ultimately used for the removal of or the formation of acetate anion in the system. So, apart from that, when we have only the methyl function attached to that, we also consider this as C1 metabolism. So, anaerobic metabolism is also can be considered as C1 metabolism. So, all these primitive enzymes which were sometimes present in our system also in human intestine, when we do not consider the presence of dioxygen molecule for degradation of any small species or any small molecule, or most of the time it is also present in rumen of cows. So, they basically do very interesting reactions, and for these we consider these two as sometimes when both of them are functioning together, that means we are talking something the assimilation of carbon dioxide or carbon monoxide for the formation of acetate or acetyl coenzyme A. We consider these two when they are coupled together, we consider them as some bifunctional enzyme also. So, this can also be considered as bifunctional. And from here we are just simply interested to know what type of metal centers or the metal ion cluster are present. So, from an inorganic chemist point of view, we are just basically focusing our attention on the metal ion cluster. So, the biologist level, some of these clusters as one C cluster and some of these as A cluster, and if we can find out how many metal centers are present there, then we try to find out that what type of reactivity it can give on those metal centers. So, the C cluster which is present in CODH, the carbon monoxide dehydrogenase, is allows organisms to use carbon monoxide as a source of energy and carbon. So, that is the metabolism. The anaerobic metabolism is therefore dependent on the use of carbon monoxide or carbon dioxide. So, they basically use for the source of energy and carbon. And sometime we will find that all these transformations, because they are also related to some electron transfer as well as proton transfers, because what we have seen in case of the hydrogenases, the reactions of hydrogenases, that they are proton coupled electron transfers. So, when we see something that the only hydrogen atom or some proton coupled electron transfer is taking place, and which are also able to synthesize at that particular point some ATP molecules. So, our basic goal to know these systems related to this iron and the nickel cluster, which we already seen in case of the hydrogenases. So, this particular C cluster is very simple, that C cluster is present in CODH, and we have seen earlier that this is a cluster of 3 iron 4 sulfur cluster. So, what we know that from the ferredoxin type of molecule, when we have certain cube like so very simply we write this motif when we have Fe4 S4, and in this particular case when we have this Fe4 S4, and here we see that what we are getting something that is Fe3 S4. So, we basically create some vacancy over here. So, if this is the iron position, so the other one is also your iron position, this is also your iron position, and that is also our iron position, and these are all inorganic sulfur, that means the sulphide sulfur. So, this we all know from the basic structure of ferredoxin molecule, 4 iron ferridox. So, if we now consider, because here we are just simply modifying the cluster. So, Fe4 S4 cluster, so we modify that cluster such that we can put some other metal center to get a heterometallic cluster. So, if we bring some metal center over here, within the cluster motif you will find that still you have 3 inorganic sulfur atoms available to bind this particular metal center. If it is say nickel, this nickel is coming over there. So, you have basically 3 sulfur groups, and they are, when they are sharing one vertex of the cube, they are basically the disposition of all these 3 sulfurs are facial disposition. So, basically we can think of something where we know that a ligand like triazacyclanonane, which is also a very unique one to give you a facial environment. So, this is also giving you some facial environment. So, this particular part, so what we basically get that one particular face providing 3 sulfur atoms which can go and bind to your the incoming metal center, which can be your nickel. So, this is like this one. So, when you have this facial environment for triazacyclanonane, which is L, consider is L, which say is coordinating to iron, and the simplest possible salt is LFeCl2 using TACN. So, we triazacyclanonane. So, this is also your TACN. So, this particular information also gives us some important clue that this immediately can go for dimerization. how these official bonds, a very labile one is a weak one for hydrolytic reactions. So, if we can go for, if it is reacting with some water, in presence of water it is also getting hydrolyzed. So, this particular unit can very easily give us something following deprotonation of these water molecules from these coordinated water. So, water will kick out the chloride functions attached to the iron center. So, basically you get the simplest possible motif, which is hydroxide-based di-iron species. So, that is the simple thing, that when you bind one particular phase and you have one metal center and some loosely bound group, say nickel chloride you are giving. So, nickel chloride immediately coming here, but on further hydrolytic reactions it can bind to some other metal center or any other cluster. So, the dimerization of the cluster is also possible through this vertex, but the simplest possible or the most critical one is how you generate this one. So, this cluster will immediately take up another position, which is your metal center. So, this C cluster is now you have, that is you have 3 iron and 4 sulfur present within the cluster, and that is bridged to a binuclear NiFe cluster. So, what I am just telling you that when you get some vacancy over here, so you get one position. So, that position can be occupied by another incoming metal center like another nickel or some other already ligand bound iron site. But if you have already you have a like hydrogenase type of NiFe cluster, so that particular cluster can also be put with that of the other existing cluster. So, that structure we will find. So, our basic interest to know what type of structure you have and how they are reacting. So, for these all these reactions, you when you get some position as nickel within the cluster, so the nickel will have very high affinity for binding to carbon monoxide, like your nickel tetracarbonyl formation or any other catalytic reaction related to carbon or any organometallic reaction related to that based on the carbon or any other carbon derived species. So, binding of carbon monoxide to nickel and water to iron. So, if it is involving some iron and if you can generate some active hydroxide function from the deprotonation of the water molecule, this water molecule which is bound to your iron center and which can give rise to some deprotonation and it generate the hydroxide ion, and that particular hydroxide ion can attack our this particular assembly, that means the nickel attached to carbon monoxide. So, that hydroxide, so in situ generated hydroxide can attack your metal carbon monoxide complex to give rise to your nickel carboxylate type of species. So, same type of reactivity pattern we can have in some other point where you can convert, we will see in case of ACS that CO is converted to COCH3 and that will be released as the corresponding carboxylate function. So, in the second step your carbon dioxide is generated and released. So, it is the interconversion of CO and CO2, but when we go, that means this particular bifunctional enzyme function, that means we take one part is CODH type, which is your isolated CODH, but the other part is ACS type. So, if you have one particular part which is your C cluster, so this C cluster will also be present in this particular one, that means all acetogenic and methanogenic bacteria, that means they are able to produce acetyl functions or acetyl, that means some important organic functions we require to go for acetylation or they can go for production of methane. So, there are some bacterial things, some methanogenic bacteria are there which can produce large amount of our that greenhouse gas. So, we are talking about consumption of one greenhouse gas like your carbon monoxide and carbon dioxide, but at the same time if your methanogenic bacteria is active, will produce another greenhouse gas which is your methane. So, in that particular case or this these two, the enzymes convert the greenhouse gas CO2 to acetyl CoA. So, this basically one particular pathway where CO2 or CO can be converted to acetyl CoA, and in another case, so another group of molecules which are again dependent on nickel, which will be utilized for some point that where the nickel center will be attached to your methyl function, that means like your methyl cobalamin, and that methyl cobalamin type of methyl nickel function can take up one hydrogen and produce your corresponding methane molecule. So, this particular structure what we were seeing from the last class, that you have the same thing, that is you have created some vacancy. This is basically your cluster, original Fe4 cluster. So, you generated some vacancy. So, once you generate the vacancy, you can have 3 sulfur groups on Fs, which is available to bind your nickel site, and sometimes this particular assembly with that of your other protein part can react and go for this particular reaction, which we already discussed in the C cluster. But now we will see that what is the corresponding function if you have A cluster. So, the detail other part of that means the ChemDraw structure for this particular thing we will see, but this is the thing what we have from the corresponding protein structure. So, this particular active site which is involving your A cluster. So, in ACS, only ACS, not coupled with CODH, you have the A cluster. So, we are just talking about 2 clusters, nothing else, one is C cluster and one is A cluster. So, if you are asked to compare that this particular C cluster or the A cluster with that of our other known clusters which are present in biology. So, one such is that your corresponding ferredoxin, then some other groups of clusters like the involving molybdenum is present in nitrogenases. So, in this particular case you have no such removal or the modification of the corresponding Cuban type of geometry. That means 4Fe4 sulfur cluster is present, and you have some thiolate bridged, that means the cysteine residue from 509. That cysteine residue from 509 is available, and that 509 cysteine residue, this residue. So, this particular cysteine residue is coming, and that is basically utilized for taking off now not only a single nickel, but 2 nickel center. One is NiP and another is NiD. NiP is nothing but your nickel center which is very close or close to that particular iron cluster, which is the proximal nickel, and D is your distal nickel. So, these could have some different functions, that when you bring not only a single nickel, but another nickel, and we are talking about some reaction where you have this methyl group. Methyl group is there which is attached to some particular center, and you have the carbon monoxide. So, what you immediately think that your one such nickel center is utilized for carbon monoxide binding, and another center is utilized or providing that particular methyl group, then these two are coupled together and forming your acetyl function. So, acetyl CoA. So, acetyl CoA generation is nothing but your individual reactivity for the metal centers for the reactants like carbon monoxide and the methyl function. So, positioning of these 2 nickel groups. So, one is there and another is there. So, you have the cysteine residues, not only this 509, you have other cysteine residues are there. So, other cysteine residues, so this is your 509, this is CYS 509, this is CYS 595, this is 97. So, they are pretty close, 95 and 97, and this is on the different side is 528. So, when this cysteine residue, that means 509 is coming into the picture, so it is basically attaching your this nickel center to the cluster. So, not the inorganic sulfide group which is responsible, because earlier in this particular case the C cluster your inorganic sulfur is coordinating to your nickel center. Now, your cysteine sulfur is responsible for not only coordination, but also for the bridging. So, you have for this coordination you have the proximal nickel, that means NiP, and for another thiolate bridge nickel, which is your corresponding distal nickel. So, again in the same fashion from the CODH, your carbon monoxide is moved through the protein tunnel basically. So, it's a long distance it can move. So, gas can move to a long distance. Then when it is approaching to your nickel side, it can form the corresponding nickel carbon monoxide complex, and the methyl group which is coming from other side, which is the corresponding iron base iron sulfur based protein, but is a corinoid structure is there. So, another ring is also present. So, methyl group is available from other center, and that methyl group is then attached to your carbon monoxide, giving again at the end point also you are generating the acetyl metal ion species. So, whatever you have, your metal center is utilized for trapping carbon monoxide, or another metal center is utilized for trapping your methyl function, and ultimately during the conversion, like it is a very simple catalytic processes. So, all the catalytic processes involving your oxidative addition and reductive elimination, that your carbon monoxide attached to the metal center is getting transformed to your acetyl function. So, finally, what we are producing? Finally, this particular coenzyme A basically binds to your acetyl and triggering the thyrolysis of the acetyl metal bound to form the acetyl CoA, because this acetyl CoA you have all the time this sulfur, again you have the thiol end. So, these thiolants, so all the time the thiolants are giving you some important reactions based on nickel. That is why the coordination chemistry related to nickel and sulfur is very interesting. So, your inorganic sulfur is utilizing for nickel binding, your cysteine sulfur is utilizing for nickel coordination, and then finally, your acetyl, which is also bound to your metal center, giving your acetyl CoA, but acetyl CoA, this is CH3CO. So, this CH3CO is attached to your CoA through sulfur end. So, sulfur end has played some important role, and if you talk about, because the stabilization of this thiolate end or the sulfur end is not so easy, even in the laboratory scale, the preparation of nickel thiolate complexes are not so easy, because for thiolate nickel is strongly oxidizing. It can oxidize the thiolate center, and it itself can reduce to nickel 1. So, here also now we see that typical drawing, which is you can have this, it is available from this ChemRay, because it is taken from this ChemRay, because all these informations and literatures are not still available in some standard reference books. So, it is directly taken from that ChemRay reference. So, this is your cluster C. So, now you can have this chem draw, why? Because this all the protein structures you cannot write in your answer book, but this you should be able to write nicely. So, you see that how much the distortion is taking place, that is very important, that you have the regular cube structure, regular cube-end structure, we call it as a very regular cube-end structure, but all these distances are not same, because you have the non-bonding sulfur-sulfur distance which will be different from that of your non-bonding, that means the diagonal Fe distance. So, you cannot have a regular square which should be distorted in some point. So, when you create some vacancy, that means this position, this site has been creating a vacancy, that means one iron center is removed, and that is also well known in iron sulfur proteins also, probably you have studied that also, the Rieske cluster. So, Rieske cluster, because occasionally they produce in all some catalytic reactions the Fe3S4 cluster. So, Fe4S4 cluster is responsible for generating our Fe3S4 cluster. So, these are still, that is the original cysteine sulfur coordination like your ferredoxin type of molecules. So, you will find that so much similarity with that of our ferredoxin type of molecules. So, when we take out this particular group, that means when you take out this iron, what we are taking out? We are taking out this cysteine sulfur also, because if you recall the Fe4S4 structure, because iron is attached over here, iron was there here, and that iron is bound to this inorganic sulfur, this inorganic sulfur and this inorganic sulfur, and this particular iron was tetrahedral in geometry. So, in all Fe4S4 molecule, all the iron environments are tetrahedral, but in this particular case, so when you take out, so this particular cysteine sulfur is the original cysteine sulfur present in the Fe4S4 cluster. So, it is moving away. So, if some mechanism is there, that means you can move out this cysteine sulfur to some distance. So, there is something and you can have some related to that type of inorganic sulfur. So, this is also your inorganic sulfur. So, if you can have some supply of inorganic sulfur and the cysteine sulfur is little bit going away from the cluster, you generate the vacancy and that vacancy can be taken up by your nickel side. And this will definitely be a distorted one. Forget about the presence of this second iron, the first one. When you have the Fe3 unit, you put the nickel, and how much distortion can be there, how much distortion can take place on the cluster structure, that you can also see, because this will definitely be a distorted structure, because whatever nickel we are putting over here, which has a complete different coordination geometry, because earlier we are trying to replace the iron site, which is a tetrahedral one, but now we are putting the nickel, and nickel has a preference particularly in sulfur environment, like what we know that the tetracyanonickelate also. So, depending upon the strength of these ligands which is coordinating to your nickel center. So, this nickel center, though you can have the corresponding environment like that of your iron, that it can also go for a tetrahedral geometry, but it is not preferring that tetrahedral geometry. Instead, it will go for a square planar geometry. So, when you have a square planar geometry, so you definitely have some distortion within the cluster. Which cluster? The cluster is now instead of Fe4, now it is NiFe3. So, you should also be able to tell or write the corresponding distortion on the NiFe3 cluster and how much it is different from the corresponding regular Fe4 structure or Fe4 cluster, which is present in ferredoxin molecule. So, this is one sort of distortion what we are getting by replacement of one iron center by nickel, and the second level of distortion is coming when we bring the second iron site over here. So, the second iron site is bringing over here, and it is already bound to one cysteine residue and one histidine residue. So, these two, nitrogen and sulfur, is basically behaving like a bidentate motif, bidentate protein ligating motif, which is available for iron coordination. And this sulfur, which was already present with that of your Fe4S4 unit, is available for coordination not to this nickel. So, this sulfur is not available for coordination to this nickel, because for the regular geometry we could thought because if it is a tetrahedral one, this sulfur will go and bind to this nickel simply, but it is not happening because already you have a planar arrangement and this sulfur is now available for this iron. So, this geometry, so you have now from this cluster structure that what type of coordination you can have. So, basically on this corresponding Fe4S4 structure, what we are putting? We are putting, that is why we made that statement, that within that Fe4S4 cluster we have created one vacancy, then we are putting one binuclear Ni iron motif like your hydrogenase motif. So, nickel iron motif which is sulfur based, which we are putting or attaching to the existing structure which has been created on vacancy by removing one iron side. So, once you put this one, so this is typically a square planar one, but this is not a square planar one, it will tending towards a tetrahedral geometry. So, this iron will have a different geometry. So, it is already bridged with this sulfur to your nickel center, and now it is attracting this sulfur. So, it is a mu 3 sulfur. So, it is providing connectivity. So, this iron is basically providing connectivity. In one end it is attaching to the cluster, and another end it is attaching to that nickel center. So, if we can consider that some electrons and the proton supply is given by your cluster structure, you have the nickel site and you have the iron site also. So, that the statement already we made that, because this iron site, which is again a low coordinated one, which is a 4 coordinated site. So, it can go for another coordination for 5 or another coordination for 6. Similarly, this nickel site, which is a square planar one, can go for its 5th coordination and the 6th coordination. So, that is why you have a low coordination site, you have generated a low coordination site, and the entire thing, the entire C cluster in CUDH, always remember that it is in protein environment. So, it is not in that it is not in some other groups or other molecules available. So, there are plenty of water molecules available. So, in all the cases where we see that if you have the bare nickel center available, any nickel salt, when you dissolve in water, it will immediately attract 6 water molecules to give you a regular octahedral geometry. But in this particular case, if we wish to have some catalytic activity based on this nickel site, your protein environment is such that it will not go for any kind of coordination from the apical site before catalysis. So, during catalysis it can go for binding the carbon monoxide or the acetyl function, but before the catalysis cycle starts, that means in the resting state. In the resting state, this nickel should be kept or should be preserved in a square planar environment. So, your protein environment should be such that, and also your coordination environment, because it is strictly a square planar one. So, your protein environment will block these positions for coming water molecules close to the nickel center to give you any pentacoordinated or hexacoordinated species based on this site, nickel site attached to this cluster. So, this is the C cluster. So, slowly one after another, if you think of from starting from your ferredoxin molecule, you should be able to draw the structure, you should be able to justify the positions, and little bit you can expect that what type of catalysis, what when we are talking about your carbon monoxide dehydrogenase, that whether your carbon monoxide can go and bind to any of the nickel site and go for the transformation. But for the A cluster, which is a completely different one and which is there, it is side by side, because we are putting these together, because side by side we can compare these two clusters at the same time. So, here this Fe4S4 cluster is intact. So, this Fe4S4 cluster we are not destroying. Here this cysteine sulfur we have taken out, and that cysteine sulfur has been utilized for further nickel coordination. Here this cysteine sulfur, this 509, still recall the number in the protein structure what we have identified. So, this cysteine residue 509, which is already attached to iron. So, this particular one can now function as a very useful bridging interaction with this proximal nickel site. So, if you have a cluster structure, forget about the right part, because this particular one is your ferredoxin molecule. So, we are studying, this is well known now as this Fe4 ferredoxin molecule, where all the 4 iron sites are attached to cysteine residues, and we are elaborating that particular cluster from one iron site. So, all these cysteine residue, this cysteine residue, this one and that one. So, all other 3 cysteine residues, if we think all these cysteine residues in terms of simple thiolate coordination. So, that thiolate coordination immediately can give us some idea that okay, this thiolate sulfur, this thiolate sulfur and that thiolate sulfur can also be useful for bridging interaction. But you should have some vacancy over here. So, within the big protein structure, because we are focusing our attention only on the cluster, only on the metal site. So, vacancy, available vacancy or available vacant site is such that only this iron can allow the bridging through its originally coordinated cysteine sulfur residue. So, you have some amount of vacancy over this particular position only, and this sulfur, this cysteine sulfur will start interacting with your proximal nickel. And what about your distal nickel? So, your distal nickel is already there and which is already bound to your both the two nitrogen sides from the polypeptide chain. So, these are amide nitrogens. So, this is the glycine residues and on the two other end you have the cysteine residues. So, cis-glycis, the tripeptide is cis-glycis. So, this tripeptide is basically providing a bidentate motif through these two nitrogen atoms. So, which is very much similar to that of your this particular cluster C, where you have this sulfur and nitrogen, one from histidine residue and another from the cysteine sulfur. So, this is your bidentate motif. So, you have this same bidentate motif, and here the bridging groups for these two sulfur, one is coming from the cluster and another is from the inorganic sulfur. But now you have some extra sulfur, the cysteine residues, one more cysteine residues and another cysteine residues from this is completing on the distal nickel a tetra coordinated environment. So, on the both two ends, so these are well known coordination chemistry motif that if you have something, if you have not only the cysteine sulfur residues which can be attached to your iron site, and that attached cysteine residue bound to your cluster molecule can show some interaction, can bind some other metal center such that it can function as a very good bridging unit, such that some heterometallic interaction can start and you can have some metal on the Fe4 cluster unit. And this on the right hand side, this right part is also very interesting motif, because this if you just block this one, these two sulfur, if you could bridge it, which is a very good macrocyclic ligand. So, is a part of the macrocyclic ligand, and most cases like this of your tripeptides. So, these tripeptides when we can have, so if they provide some groups like 2 nitrogen and 2 sulfur. So, basically it is a N2S2 acyclic, this part is only acyclic. So, rest is cyclic. So, N2S2 groups are available, and these sulfur groups, they are thiolate sulfur. If you make them SR, SR or S-methyl or S-methyl, this will not be available for any bridging function. So, that gives us to large number of coordination compounds also, and this sort of chemistry is well known before the discovery of these molecules. So, these are very recent identification, but once you know the typical coordination chemistry related to this macrocyclic motif, you should be able to tell, because immediately we can have some time during the structure solution of the protein, we can very immediate difficulty whether this is S or SR or S-methyl. So, immediately since the nickel will be identified first, because all the heavy atoms, all the metal centers will be identified first. So, when you identified nickel and these two nickel centers, you can immediately find out, even if you do not solve the structure, because large number of extra structures people have solved so far for this thing to get a accurate structure. So, accurate bond distances and all. So, when you determine these two nickel positions or find out this nickel position, these two nickel distances are less. So, this is basically a N2S2 diamond structure. So, both of them are bridged by the thiolate sulfur. So, this has a very short distance. So, immediately that will tell you no other interaction is coming from like your other L group, that means other monodentate ligand, it will be only bridged by these two sulfur. So, during the catalysis, so what you have not only, so this iron site is not, instead of iron here, you have now one nickel site. So, both of them are now nickel. So, during the formation of this acetyl function, we will get something, that means these two are very common all the time, the CC bond formation, that means is a simple one, this acetyl group formation, the attack of the carbon monoxide to your methyl group, and the CS bond formation when your sulfur or thiol end of the coenzyme is attaching to your acetyl function. So, this can be a little bit complicated one, but once you realize the structure. So, this is now we are simply focusing our attention on the metal centers, because the metal centers are available or metal centers are responsible for this catalysis. So, how the positioning. So, basically is a very complex in terms of your metal complex. So, in terms of a metal complex is a very complex architecture. One end you have the cluster unit, but that cluster unit is required for holding this nickel, and this nickel site what we can have this in the low coordination site, because already what we have shown in the previous structure, that you have L. So, one loosely bound L is there, and you see when L is present, this nickel, the proximal nickel is in its preferred coordination geometry, that coordination number is 4. Otherwise, it can simply go away to leaving behind your nickel in tri-coordinated form. So, when they are in the low coordination, so immediately you can think of that, like in the copper center also, the copper or nickel in a low coordination number, that is the coordination number 2 or 3, you can go down, that means you can reduce that particular site very easily to nickel 1. So, when it is in the low coordination site, that means already it is bridged by 2 cysteine sulfur residues, and so it is typically in sulfur environment. So, typically in sulfur environment, and what we find this particular case, that means in this particular case when you go for this, and if you have if you do not get the corresponding electron transfer to that particular site. So, this particular nickel site is, even if it is forming or immediately it is reacting with a nickel in bivalent state, it will immediately go for reduction, because you have typically cysteine sulfur residues, which can provide huge amount of charge density to the metal site. So, this particular in case of copper also, because what you can have, if you allow it to react with any nickel salt, and if your nickel 2 plus salt is oxidizing in nature, it can immediately go for oxidation of cysteine sulfur or the thiol sulfur to give you disulfide. Your byproduct would be disulfide, and your metal center will be reduced in the +1 oxidation state, if you are unable to generate nickel in 0 oxidation state. So, always you will have the tendency, because even if you do not have the nickel in the bivalent state, but if you have the thiol coordination. So, thiol group, they are basically the sacrificial group, the thiol groups themselves will destroy, and that thiol functions will destroy, and that destruction will give us some important thing, that this will be reduced to nickel 1. Why we are talking about the formation of nickel 1 in this particular one, that means the proximal nickel. So, this proximal nickel is our catalytic site, which in the low coordination, its coordination number is 3 compared to your distal nickel, which is your nickel 2 plus, because it is already in nitrogen environment, it is not that it is in all sulfur environment. So, if you compare the coordination environment of these two, this nickel site will be available for this particular reaction, which is coupled with CODH. So, what we are talking over here is the center which is available, the cluster available on the ACS, and this particular part is dependent on CODH, because carbon monoxide dehydrogenase is used there, and which is giving us that in situ generated carbon monoxide, because we do not have carbon monoxide there, we have only carbon dioxide. So, during this conversion, the CODH is active, and that CODH is reacting with your carbon dioxide, forming your carbon monoxide, and that is why in the previous slide I have shown you that I wrote you also that there is a channel, and through that channel your movement of this gas is taking place. So, the gas molecules are moving through the biological channel or the protein channel, and they are basically going and approaching to the nickel site. So, is immediately nickel in the low oxidation state you have, and this immediately goes for the attachment of carbon monoxide. So, for the work for several years people are trying to identify these things, but initially we know the activity, the catalysis we know, that means identified that this particular enzyme and the other enzyme contains iron, they contain nickel also. So, when you identify that nickel center is present and is responsible for your CODH activity or ACS activity, so immediately people are trying to look at that whether, because they have the previous knowledge for the several years, say more than 100 years knowledge of nickel tetracarbonyl. So, that particular information they can have, the nickel can bind in the zero oxidation state in nickel tetracarbonyl. So, carbon monoxide can interact with our nickel. So, is a nickel carbonyl species can be generated. So, the people take the help of the spectroscopic technique, is a very useful spectroscopic technique is our IR. So, FT-IR would be useful, because when this particular part, that means when this particular center the CODH is generating carbon monoxide, and this carbon monoxide is in the free state, in the gaseous state. So, if we are able to take out this particular sample, so gas sample can also be analyzed in a different cell holder for IR. So, gas can be analyzed. So, this CO has a different stretching frequency compared to the CO which is bound to your nickel 1. So, monovalent nickel is bound, which has a characteristic 1996 centimeter inverse stretching frequency, that immediately tells us that you have carbon monoxide bound to your nickel site. But the ambiguity for the nickel site, whether it is a bivalent nickel or monovalent nickel, because your nearby nickel site is bivalent. So, that ambiguity you should have. So, what you can do? You can do now the EPR spectra. So, EPR spectra for this nickel, this is your 3D9 system, and this is also your 3D9 system, both are EPR active, but in one particular case you have bound carbon monoxide, in another case it is not. So, if you side by side, if you compare these two EPR spectra for this particular species and that, you will find that this will have some characteristic corresponding nickel spectra for G values of 2.08 and 2.028. So, is basically when two G values are available, we call it as a axially distorted spectrum. So, basically is axially distorted. So, is one axis distortion is axial distortion, the other one is your rhombic distortion, 3 axis distortion. So, all the 3 are different, x, y, z are different. So, you can have 3 G values. So, depending upon the symmetry or the environment of the system, you can have, and this particular value is very much characteristic, which is for your nickel in +1 state and bound carbon monoxide. So, you have this particular carbon monoxide. So, we are happy to have a system where your nickel is bound to your carbon monoxide, and now we have to provide your methyl group. So, anything related to that, that means your transmethylation reaction, if it is based on your methyl cobalamin function. So, your methyl cobalamin, so any cobalt 3 site, so methyl cobalamin is the trivalent cobalt, and that particular, so your methyl group is there. So, basically they are providing the methyl group. So, they are providing your methyl function to your nickel site. So, initially, because still there is debate about the function of these two sites, that means whether this one site is responsible for carbon monoxide binding, people proposed also that the other nickel site is responsible for binding of methyl group, but it is not that, because this is a simple bivalent nickel site in N2S2 environment. So, it will not show any kind of that activity that binding of methyl function to it. So, this nickel site when it binds, so it is transferring this particular methyl function from that cobalt center, and you get that particular methyl nickel function. So, it is very weaker one, because you immediately after this monovalent nickel site which is EPR active, in this particular case you can establish the corresponding oxidation state of the nickel for this any internal electron transfer, because you have this in the bivalent state, because both the two are in bivalent state and they are EPR inactive. So, this particular species in the catalytic cycle will be EPR inactive. So, most of these type of biological catalytic cycle will rely on not only the IR spectra, but also the EPR. Initially, we should be able to identify which particular species is EPR active and whether that species is giving rise to the generation of your corresponding product. That means, how the product is generating from there. So, this is EPR active. So, when you react this with your methyl cobalamin, so some species is generating which will be EPR inactive. So, it is a very simple one. People have tried all these model and all these reactions from there. It is basically based on the model studies. Even you can do also, because even we can have this very useful thing that the vitamin B12, the modeling of vitamin B12 is very easy to do. You can take DMG, dimethyl glyoxime. So, 2 of the dimethyl glyoxime can bound to any cobalt center. So, it is basically giving you the in-plane binding, and that in-plane binding is very much similar to your corine binding. So, cobalt DMG complex, so cobalt twice DMG. So, that cobalt DMG has 2 protons, so one is going for deprotonation. So, this is very much similar to that of our vitamin B12. So, if you have the vitamin B12 where you have the cobalt center, and we are talking about the binding of the methyl group on the cobalt center. So, this particular one is also is a very simple reaction on the cobalt DMGH, that what you have to do, you have go for reduction reaction from there. So, sometimes sodium borohydride is fine, and some source for methyl. So, this will generate bound to methyl, very easy to make. In the laboratory people have tried that thing. So, this cobalt which is bound to 2 DMG, so DMG is this is the oxime functions. So, you have the methyl function, and sometime we need for the very useful reaction on this cobalt center for the attachment of the methyl, we require some base. So, for that reason we simply use pyridine. So, the base is now your pyridine. So, this particular thing, that means if the reactive pattern is like this, so model studies can be made, because in the laboratory you can make. So, whatever we just basically go for this sort of model study, that you make this, and our challenge is for that is you have already a nickel monovalent nickel bound to a carbon monoxide, and we want to supply that the nickel CO Me. So, this was the information we know, that there is a nickel catalyzed reaction. So, if you are able to have some other type of this particular environment like that of your cobalt environment, so some useful or good ligand environment you can make, such that you are able to stabilize nickel in the monovalent state, and you just go for the corresponding carbon monoxide compound. So, you analyze it by recording the IR spectra, then you use this so as the source for your methyl function. So, then whether in the model compound also you are able to transform this particular nickel carbonyl compound, nickel carbon monoxide compound, the nickel carbonyl compound can be transformed from this. So, it can be has some transient existence, it may not be a very stable one, immediately it can go for the corresponding acetyl function. So, this particular attachment, so you just see that whether, because what you are getting, you are transferring this over here. So, whatever you are leaving behind with this compound, which will be the corresponding cobalt ACO compound. So, what you are getting from there, that means now you have to identify that some spectroscopic technique or any other analytical technique you have to identify that instead of your COMe unit, now you have the cobalt aqua unit, cobalt water unit, and then that methyl group is moved to your nickel side. So, that immediately from the informations what we can have, that the model studies of the reaction between methyl cobalamin and nickel 1 plus any macrocycle now. So, the nickel is there. So, this particular nickel is the protein is stabilizing this particular nickel site, but here any kind of macrocycle can also do the same reaction. So, like your cysteine sulfur, because the last molecule what we will see next day, that the nickel in a macrocyclic environment can also be stabilized in the lower oxidation state. It is not that the whatever macrocyclic compound you make, that all be stabilized in the nickel plus 2 oxidation state. So, it can be stabilized in the plus 1 oxidation. So, if you have a nickel macrocycle, which can also tell us that you can transfer this methyl radical from this particular cobalt function or the cobalt center to the nickel center. So, at this point, because both the ingredients or both the substrate, the methyl substrate and the carbon monoxide functions were attaching to the nickel. So, is very similar to that of some catalytic processes like the Monsanto process for the acetate preparation or the acetic acid synthesis, the industrial preparations what we see. But that time that industrial thing is known, but this particular detailed information about the biological thing was not known. But nature is doing in the same fashion, the same metal center is utilizing or holding these two, and at this point you have the CC bond formation on the nickel. So, that is why you can think of something the nickel based catalysis, and you can think of, since it is coming out from the biology. So, it is the bioorganometallic species. So, bioorganometallic intermediate is there. So, protein is also doing the same function what we are utilizing for big industrial processes, that means the catalytic processes. So, the simplest thing is that like you are using palladium and all other metal center, rhodium and all the Wilkinson catalyst and all. So, you have the center generated, the center is reduced and you attach these two groups. Now, this particular one you have the corresponding CC bond formation, and after this CC bond formation what you can have, you have this is the deprotonated acetyl coenzyme. This originally it is SH. So, at this point it is the SH is a deprotonated one. So, deprotonated acetyl coenzyme A is reacting with this one and taking up this acetyl function to give you acetyl coenzyme A. So, at this point what we are giving you this, we are getting that is CC bond formation, and here you have the CC bond, CS bond formation. So, these are the two basic steps. That means, at one point you are generating the CC bond and another point you are generating the CS bond. So, once it is giving, so you are just going back that. So, what basically we learned from this particular thing, that you have the metal center catalysis and it is involving your bioorganometallic intermediates. So, these are the basic things. Okay, thank you.